I'm originally from Brazil. Since I was a kid, I was really playing around with science kits and trying to understand like the basics of life. I was really amazed by the universe. I mean, try to understand the entire universe. It was only then when I get to know more about the human brain that I realized that, whoa, the brain is the last frontier. The number of cells that you have is something like amazing. You carry more than 1 billion neurons in your brain. The activity of those neurons generate what you think. And to me, that was something amazing. I mean, these fundamental questions, who am I? What I'm doing here? This all came to life when you think about the human brain. To me, that was the complexity was the major attraction to me. If you understand the brain, you can understand the whole universe. I got a Ph.D. from genetics at University of São Paulo in Brazil. We have this program that allows you to go abroad, and I went to a lab in Harvard in Boston, and I was very surprised the way the science was made in US. I think most of you probably don't know how science is actually done outside the US, but in Brazil it was very, the support for science is not that great. Usually it's very small budgets, and you have to think about projects that are not too ambitious, so you can finish in a decent amount of time. If you order an antibody, you're gonna have that antibody 6 months after you order. So when I came to US, I realized that, the availability of reagents and materials was it's a different story. It was quite good. I mean, it's right there. What keeps you from not doing the experiments is basically ideas. If you are short on ideas, then you don't do the experiments. But if you have plenty of ideas, you can keep doing science all the time. And I felt like a kid in a playground, just playing with these new toys. There are very important colleagues, neuroscientists, who made like fundamental major discoveries here. And they have something that I don't see in any other place, which is a lack of an ego. So you can talk to these guys, interact and collaborate in a very easy, friendly way. Originally, the first interest was really to understand what makes the human brain so unique compared to the other primates. One aspect of the human brain that's very different from other primates, it is your social brain, your ability to interact with different people. One way to really attack that question was to find instances of or diseases or mutations that makes the social brain malfunctioning. Autism would be a good way to try to understand the human social brain. You can study autistic individuals by trying to scan their brains or EEGs, but that was not enough. We want to be able to do experiments with their cells, with their neurons. Shinya Yamanaka showed us that you can actually reprogram one cell type into another, and he did that by changing the fate of a fibroblast, a skin cell, back into this embryonic-like state. And from that stage, that naive state, you can create all the different cell types in the body, including brain cells. So the first experiment was, okay, let's use stem cells to model autism. We will collect somatic cells from a group of autistic people, somatic cells from a group of control people. We will reprogram the cells back to this embryonic state and make neurons from them. And let's see if those neurons behave or if they are if they have any morphometric differences. At first, when you look at them, they don't seem that different. But it was only when you start zooming in on the neuronal branches of the process and counting the number of spines and the number of synapses that we realized that there's something wrong with those neurons. They don't make as many synapses, they don't have as many spines. We also follow up with a more functional difference. So if they do have less synapses, perhaps the neurons would have electrophysiological properties that distinguish them, and turns out to be true. There is a clear difference between neurons that were derived from the autistic group and from the control group. There are two main questions in the lab. One is try to understand what is the causes, how to link the genetic information that we can get from these patients. And sometimes there are more than like 20 mutations from that specific individuals. How those 20 mutation causes those differences, the lower number of synapses, for instance. And we take advantage of genome editing, so we can manipulate those mutations in the pluripotent cells and then make neurons with less or more mutations and see how they causes the phenotypic differences. The other thing is, okay, we are not so interested on the causes, but we want to be able to revert that, making the autistic-derived neurons close to the normal ones with pharmacological treatments. I think it gives you the scientific method perspective. So if you have an idea, if you want to test this idea, what are the proper controls that you need to do? How would you convince someone that your answer is the correct one? To me, the best students are the ones that have an intrinsic motivation. They don't need me to keep pushing them to do the experiments, right? They would do by their own because they are very curious. Find a good mentor. Find someone who really excites you. Find a research that makes you stay in the lab for long hours. So, I mean, being able to find this person or this lab, to me, is quite essential.